Hello, and welcome to my presentation for Douglas Richmond Soil Insect Ecology for 393. These past weeks, I was shadowing under Professor Richmond's lab, which is a research and extension laboratory, and the social grant challenge they are trying to address is climate change and food security. I attended laboratory joint meetings with Professor Long's lab and interviewed several professors' lab members like Avila Arez Helena, Ryan Beard, and Connor Sturr. Start off with Professor Richmond is a teacher and extension officer that creates projects for graduated students to conduct. And so he focuses on turf grass system as the main model to conduct many of these experiments. He has a bachelor's in biology, a master's in entomology, and he received his PhD by studying these turf grass systems and their pests. So he knows the ins and outs. And as you can see, the bottom picture is the area where he does most of his experiments. The top picture is the pest that he's trying to collect and gather as much data on. So he aids in projects. So he helps all the lab members like Avila's microbiology and her carbon nitrogen cycling, Ryan and his project with prairie restoration pollinators, and Connor with his mint and scarab pests. During the summer, he helps industries with their insecticide products to manage soil insects that are making their crops become infested. So they're trying to figure out management ways to protect their crops. Next is Avila Arez Helena. She's a microbiologist who researches the relationship between the soil and Japanese beetle, and her reasons for exploring this area is because they have one generation per year and live in the soil for 10 months out of the year, and they only come in June and July. There's not a lot of information about these microcommunities in and out of the soil, and greenhouse gas emissions evolve around the soil and the larvae within the soil. Her project is to determine the influence of GAB in the soil, focusing on the microbes found in the soil, in the midgut, and in the hindgut, as the diagram on the right shows all the microcommunities within each of these areas. Soil carbon dynamics and carbon and nitrogen cycling in the soil, and lastly, understand the sequences of affiliations with JB. Her goal is to understand roots of grass and researching the shotgun mechanism with JB, and potentially studying from DNA, what she's saying right now, to RNA and finally to proteins. And this is to help detect JB without destructive sampling in gut microbes. Next is Ryan Beard. He's a first-year graduate student who's working on renovating no-play zones on golf courses, like the Indiana Native Prairie Conservation areas. So he's studying drill seeding versus broadcast seeding, fall versus spring seeding, and lastly studying plant covers and plant establishments and pollinator abundance and diversity. There's a lot going on there, but he's studying these areas for the summer and the fall. And his goal is to create a game plan for golf course superintendents by creating a timeline for them for their pests and when they occur. Next is Connor Sturr. He received his degree in entomology at Michigan State University and worked with the Asiatic garden beetle in corn, and he came to Purdue to study AGB in mint. So he discussed his first chapter of his thesis to figure out the maximum precision and minimize effort by researching sampling intensities. And he's comparing the cutter and the quadrats to see which is better. And he eventually said the quadrats were better for sampling methods. And he's also figuring out an approach for you to use random or systematic sampling. And with the simulated data he used, he tested the approaches and concluded that random sampling is better because it's not unbiased. Lastly, he discussed chapter 2 of his thesis, which is develop a damage threshold, and he's finding relationships between grub density and above and below ground biomass, and analyzing the importance of fall and spring to see which one is better for density and damage, concluded that fall was better for both, and he found there's an interrelation between field and season and year. So in the end, these past weeks, being a part of their lab meetings and understanding these individual projects has all involved different aspects of soil insect ecology. They answer climate change and food security challenge by helping farmers and golf course workers about what approaches to take in handling these pests. I've learned that turfgrass is a great model to look at these pests for students trying to understand these concepts like me of soil insect ecology. Thank you.